The Entner-Doudoroff pathway of sugar catabolism is present in many archaea, bacteria, and eukaryotes. There are several variants of the pathway, but generally it looks as shown on the slide. If we start from glucose, the first reaction is identical to that of many other pathways that handle sugars and is glucose phosphorylation by glucokinase to form glucose 6-phosphate. It is oxidized to 6-phosphogluconolotone and then hydrolyzed to 6-phosphogluconate by dehydrogenase and lactonase respectively. Looks like first oxidative reactions of pentosophosphate pathway. Other two reactions are unique to the ED pathway. It's a conversion of 6-phosphogluconate to ketodeoxyphosphogluconate by phosphogluconate dehydratase, and its breakdown to pyruvate and glyceraldehyde 3-phosphate by ketodeoxyphosphogluconate aldolase. One molecule of glyceraldehyde 3-phosphate is processed like in the pay-off phase of classical Embden-Mergov-Parnas glycolysis to give another molecule of pyruvate, ATP molecules, and NADH. It can be seen that apart from the EMP pathway or classical glycolysis that gives 2 ATP and 2 NADH, the ED pathway gives 1 ATP, 1 NADH, and 1 NADPH per 1 glucose in total. One molecule of ATP is used for activation. 2 molecules produced in the pay-off phase. Namely, it would seem that the EMP pathway is strictly superior, yielding twice as much ATP as the ED pathway. This begs the question, why is the ED pathway common? One assumption is that the ED pathway is just a preserved vestige of carbohydrate metabolism, just a historical artifact. Think about this. Most glycolytic pathways converge at the level of glyceraldehyde 3-phosphate, which is then converted by a common trunk pathway of enzymes to pyruvate. This is the pay-off phase or lower glycolysis. It is a common and ancient part of both the EMP and ED pathways. This trunk pathway appears to be present in most species when preceding part of glucose metabolism may vary even within the ED pathway. For example, halophiles possess a modified ED pathway in which the intermediates are not phosphorylated until the level of ketodeoxygluconate. It is phosphorylated to ketodeoxyphosphogluconate. Aldolase then cleaves the ketodeoxyphosphogluconate into pyruvate and glyceraldehyde 3-phosphate. The last one is metabolized to pyruvate by the trunk pathway enzymes. Thermophiles have the same modified Entner-Doudoroff pathway as the halophiles, but here the 2-keto-3-deoxygluconate is cleaved into pyruvate and glyceraldehyde without any phosphorylation step. The glyceraldehyde is converted into glycerate, which is in turn phosphorylated to 2-phosphoglycerate by glycerate kinase. Enolase and pyruvate kinase complete the pathway. These two enzymes are the only glycolytic enzymes present in the thermophiles. If the archaea do indeed retain features of the earliest form of life, one possible scenario is that a very early form of glucose catabolism corresponded to a mainly non-phosphorylated ED sequence that yield only a single molecule of ATP for every glucose degraded. In this case, the ED pathway in archaea would represent a vestige of earlier glycolytic pathways. Pyruvate kinase and enolase are the most ancient glycolytic enzymes here. The ubiquity of the ED pathway, however, suggests that it is of far greater importance. So what would be the role of the pathway? It is often said that the primary difference between the EMP and the ED pathways is energetic. The ED pathway yields less ATP and thus have more exergonic reactions than the EMP pathway. Moreover, kinetic analysis carried out in some works says that the ED pathway requires several-fold less enzymatic proteins than the EMP pathway to metabolize the same amount of glucose per second. Of course, cells can synthesize more enzymes to make the flux via the EMP pathway better, but at what cost? Increased protein production is thought to impose a significant burden on cells by occupying ribosomes, consuming cellular building blocks, and increasing misfolding. So we can suppose that the ED pathway is a trade-off between glycolytic ATP yield and protein cost. Under this hypothesis, organisms that depends on glycolysis for ATP production, for example, fermentative anaerobes, will tend to use the EMP pathway, while organisms with alternative sources of ATP, such as aerobes, will favor the ED pathway. Some studies support this hypothesis. For example, cyanobacteria, algae, moss, fern, in general, are nutrient limited rather than ATP limited. They even possess non-energy conserving bypasses in their respiratory chains that would enable the oxidation of carbohydrates for a biosynthetic purpose. ATP can be oversupplied. Calvin cycle, oxidative part of pentosophosphate pathway, and the EMP pathway share intermediates. Thus, the simultaneous operation would result in futile cycles, decrease carbon dioxide fixation, and enhanced oxidative stress. Thus, that's why the ED pathway is physiologically significant, as it does not produce the futile cycles, especially under mixotrophic conditions when light and glucose are provided and under autotrophic condition in a day-night cycle. The production of NADPH by the ED pathway may be particularly significant. It is required for anabolic enzyme reaction, but also supplies reductants for oxidative stress protection. A notable exception is the Zymomonas mobilus, an obligate anaerobe used to brew African palm wine. Zymomonas mobilus is likely adapted to grow at very, very high sugar concentrations. Its ED pathway is constitutive. Zymomonas lack phosphofructokinase and thus does not have the EMP pathway. Its pyruvate is converted to aldehyde and ethanol. Another example is Escherichia coli, which is capable of both pathways and grows quickly in aerobic environment. It tends to use the EMP pathway. However, its ED pathway is inducible and may allow colonization of mucus. It serves as a funnel receiving metabolites derived from the catabolism of several sugar acids, including gluconate, iodinate, glucuronate, galacturonate. Each of these compounds is found in nature and in several cases is known to be present in significant concentration in the mucous layer and is also available from degraded food in the intestine. Interestingly, that colonization of intestine by Vibrio cholera is also accompanied by the induction of the ED pathway enzymes and virulence genes under these poorly oxygenated conditions. Pyruvate thus obtained can be fermented or oxidized to acetyl coenzyme A and then in the citric acid cycle. Next examples demonstrate that the ED pathway is involved in anabolic metabolism. Some species, such as Pseudomonas aeruginosa or Campylobacter, do not have phosphofructokinase. They have the cyclic ED pathway. As suggested, the physiological rationale for this might be related to an enhanced glyphosate supply. This would be particularly important for the formation of capsule polysaccharide or lipopolysaccharide expressed on mucosal gram-negative bacteria. The ribulose monophosphate pathway is used for formaldehyde assimilation in a number of obligate and facultative methylotrophs. The formation of fructose 6-phosphate from formaldehyde and ribulose 5-phosphate occurs first. Fructose 6-phosphate is in several reactions those that involve the ED pathway enzymes is converted to glyceraldehyde 3-phosphate. Part of glyceraldehyde 3-phosphate is then recycled to ribulose 5-phosphate. Thus, we have described some possible functions of ED pathway. Thank you for your attention.